Okay, moving to the next topic, statistics from clinical science. Things you need to remember: mean, the sum of all values divided by the number of values. Very easy. It's basically the average of everything. Median is the medial value in the list of numbers. To find the median, your numbers must be listed in numerical order from smallest to largest, like this, and then the medial one would be the median. And mode is the value that occurs most often. If no number in the list is repeated, then there is no mode for the list. So if there is repetition, that's mode. Okay, skewness. In probability theory and statistics, skewness is a measure of the asymmetry of the probability distribution of a real valued random variable and above its mean. Skewness of a value can be positive or negative or undefined. So here it's symmetric, skewed left negatively, so it's more than the mean, and skewed positively is less than the mean. So normal symmetrical distribution is mean, median, mode are same. Positively skewed distribution is mean is more than median and mode. This is the positive skewed. Mean is more than median and mode, and negatively skewed is mean is less than median then less than mode. To remember the above, note how they are alphabetical order. Think forward with this and backward with this. So for positive, forward; for negative, backward. Okay, so some of the things may not make much sense, but have to remember. Okay, confidence interval describes a range of value around the mean, an odds ratio, a p-value or a standard deviation within which the true value lies. Okay, so mean plus 2 into standard error. Normal distribution is also known as Gaussian distribution or bell-shaped distribution. It describes the spread of many biological and clinical measurements. Properties of the normal distribution: symmetrical is equals to mean equals to median equals to mode, and which means 68.3% of the value lie within the one standard deviation of the mean. So these values are pretty important. Sometimes in the exams these kind of questions come. 68.3% of value lie within the one standard deviation of the mean, and 95.4% within 2 standard deviation of the mean, and 99.7% within 3 standard deviation of the mean. This is often reversed, so that within 1.96 standard deviation of the mean lies 95% single value. So the range of the mean is minus 1.96 into standard deviation to the mean is called the confidence interval. If a repeat sample of 100 observations are taken from the same age group, 95 of them would be expected to lie within that range. So 1.96 standard deviation is 95%. That's what you have to remember. Now what's standard deviation? It represents the average difference each observation in a sample from represents the average difference each observation in a sample lies from the sample mean. From the mean, how much difference? SD is square root of variance. Example: a study is performed to find the normal reference range for IgE levels in adults. Assuming IgE levels follow normal distribution, what percentage of adults will have IgE level above 2 standard mean from the mean? So we already know 2 standard deviation, 95.4%. So for normal distributed data, 95.4% lie within 2 standard deviation of the mean, leaving 4.6% outside the range. So therefore 2.3% of value will be higher and 2.3% value will be lower. So 95.4% is within this 2 standard deviation. So on both sides there will be 2.3, 2.3, which is 4.6. This figure is sometimes approximately 2.5%. Correlation and linear regression both are used when you want to know whether one measurement is variable. One measurement variable is associated with other measurement variable. You want to measure the strength of these equations R square, or you want an equation that describes the relation between two variables. So correlation is basically you do this mathematics. These charts are there. May be plotted on a scatter plot. For example, age may be marked on the x-axis and systolic PP on the y-axis. So as age increases, systolic PP increases, something like that. Positive correlation. The correlation coefficient, sometimes referred to as Pearson's product moment, indicates how closely the point lies to a line drawn through the plotted data. It's denoted by the value R, which may lie between -1 and 1. R1 is equal to strong positive correlation like this. Systolic BP increases with age. R0 is no correlation, like this, no correlation. There's no relation between systolic BP and age. And R-1 is strong negative correlation. So systolic BP decreases with age, this one. So okay, so this is like basically Pearson's product moment, if you remember this. while correlation coefficients give information about how one variable may increase or decrease with another. They do not give information about how much the variable will change. They also do not provide information on cause and effect. Linear regression is also easier. Just it's also a relation between two things. In contrast to correlation, efficient linear regression may be used to predict how much one variable changes when a second variable is changed. The regression equation will be given as this: y is the variable being calculated, a is the intercept value when x equals to 0, b is the slope of line of regression. Simply put, how many y changes for a given x. X is the second variable. Okay, comes a topic which is very common in the exam: significance test. So significance test, it plays a key role in experiments. They allow researchers to determine whether their data supports or rejects the null hypothesis, and consequently whether they can accept the alternative hypothesis. A null hypothesis is what states that two treatments are equally effective, so and is hence negatively phrased. So null hypothesis, there is no difference between two treatments. A significance test uses the sample data to assess how likely the null hypothesis is to be correct. The p null is as in nil, no difference. The p value is the probability of obtaining at least a result at least as extreme as the one that was actually observed, assuming that null hypothesis is true. Okay. Probability of obtaining a result at least as extreme as the one that was actually observed. The null hypothesis is rejected if the p value is smaller than or equal to the significance level. So it's important. So the null hypothesis will be rejected if p value is smaller than or equal to the significance level. The alternative hypothesis is the opposite of the null hypothesis. There is a difference in the two treatments. Okay, type of error. So a lot of questions come regarding this. The type of error will be may occur when testing the null hypothesis. Type 1 is the null hypothesis is rejected when it is true, showing it's an error, showing a difference between two groups when it doesn't exist, significance level. So it's showing some difference but it doesn't exist, so it's although null hypothesis is true, it's rejected. And type 2 is null hypothesis is accepted when it is false, so it's failing to spot a difference when one really exists. Okay, then comes the power. The power of the study is probability of correctly rejecting the null hypothesis when it is false. Power equals to 1 minus probability of type 2 error, and can be increased by increasing the sample size. So yeah. So rejecting the null hypothesis when it is false. So when it's false, it's better to reject. So that's good. Types of significance test. That's also very common question in part 1. Depends on whether the data is parametric, something that can be measured, or normally distributed, or non-parametric. So it's measurable or not. Paired data refers to data obtained from a single group of patients. So paired data is single group of patients. Example, measurement before and after intervention. So these are all paired. Same patient. Unpaired data comes from two different groups of patients comparing response to different interventions in two groups. So parametric test examples are measurable ones, normally distributed. Students t-test, paired or unpaired. Pearson product moment coefficient correlation. So these are like parametric and okay. And non-parametric are which cannot be measured or not normally distributed, like Mann-Whitney unpaired data. Wilcoxon matched pairs compare two set of observation on a single sample and is paired. Chi-square used to compare proportion or percentage. Spearman-Kendall correlation. McNamar's test is used on nominal data to determine whether the row and column marginal frequencies are equal. Correlation parametric. So correlation would be Pearson's coefficient, and non-parametric, not measurable, is Spearman's coefficient. So correlation, these two are there: Pearson, Spearman. Standard error of the mean is standard deviation divided by square root of number of patients. Here, so standard error of the mean is basically is a measure of the spread expected for the mean of observations. How accurate the calculated sample mean is from the true population mean. So this is equals to S by square root of n, where S is the standard deviation and n is the sample size. Standard error gets smaller as the sample size increases. The confidence interval for the mean can be calculated in a way, calculated a similar way to that for single observation. 95% confidence interval mean equals to mean minus 1.96 into Cm. No need to remember that much. Okay, incidence and prevalence. The two terms are used to describe the frequency of a condition in a population. So the incidence is the number of new cases per population in a given time period. So new cases. And for example, if condition X has caused 40 new cases over the past 12 months per thousand of the population of this, the annual incidence is 0.04 or 4%. The prevalence is the total number of cases per population at a particular point in time. Total number. The new cases. For example, imagine a questionnaire is sent to 2,500 adults asking them how much they weigh. If from this sample population of 500 of the adults were obese, then the prevalence obesity would be 20%. Prevalence is then it's already there. Total number. The incidence is the total new cases right now. So relationship is prevalence equals to incidence into duration. Incidence into duration. So in chronic disease, the prevalence is much greater than the incidence, and in acute disease, the prevalence and incidence are similar. For conditions as common cold, incidence may be greater than the prevalence. So incidence is like what's happening now, new case. Prevalence is like overall total number. So in chronic disease, prevalence is more than incidence. Okay, probabilities. Are the probabilities of the presence of a condition before and after a diagnostic test. Post-test probability in turn can be positive or negative depending on whether the test falls out as a positive test or negative test, respectively. In some cases, it's used for the probability of developing the condition of interest in the future. Pre-test probability, prevalence of condition, the proportion of people with the target disorder in the population at risk at a specific time or time interval. For example, the prevalence of rheumatoid arthritis in the UK is 1%. Post-test probability, the proportion of patients with that particular test result who have the target disorder. Post-test odds divided by 1 plus post-test odds. Probabilities are usually given as percentage. Odds can have any value from 0 to infinity, and they represent a ratio of desired outcomes versus the field. Odds are a ratio and can be given in two different ways: odds in favor, odds against. Just reading. So coming to the topic now, odds. So odds are defined as the probability that an event will occur divided by the probability that an event will not occur. So occur versus not occur. What are the odds that this will happen? That's how you say it. Pre-test odds, the odds that the patient has the target disorder before the test is carried out. Pre-test odds is equals to pre-test probability divided by 1 minus pre-test probability. And post-test odds, the odds that the patient has the target disorder after the test is carried out. So post-test odds equals to pre-test odds into likelihood ratio. And the likelihood ratio for the positive test result is sensitivity divided by 1 minus specificity. We'll come into that later. So odds ratio may be defined as the ratio of the odds of a particular outcome with experimental treatment and that of control. So this one is odds and this is odds ratio. These are different. Odds ratio may be defined, told you already. Odds, remember, a ratio of the number of people who incur a particular outcome to the number of people who do not incur the outcome, not to the number of people do and do not. Okay, for example, if you look at a trial comparing the use of paracetamol for dysmenorrhea, the total number of patients paracetamol achieved pain relief versus pain relief. 60 patients who took paracetamol achieved 40 of them received pain relief, and 90-30. The odds of achieving significant pain relief with paracetamol is 40 divided by 20, because 20 did not receive pain relief. So yeah, equals to 2. 40 patients who achieved, 20 patients did not achieve. And the odds of achieving significant pain relief with placebo is 0.5, because 30 who received and 60 who did not receive. Therefore, the odds ratio is 2 by 0.5. The odds ratio is 2 by 0.5 is 4. So it's very important. Good example for the exam. Relative risk. The relative risk is the ratio of the risk in the experimental group, experimental event rate, to risk in the control group, control event rate. CER is the rate at which events occur in the control group, and ER is the rate at which events occur in the experimental group. Control event rate is number who had particular outcome with the control, total number who had control, and experimental number who had particular outcome with the intervention, and who had the intervention. For example, if you look at a trial comparing the use of paracetamol for dysmenorrhea compared to placebo, we may get the following results: a total number of patients and experience significant pain, and placebo experience. Okay, so let's go through it again. The rate at which events occur in the control group, rate at which in the experimental group. So for experimental event rate, it's like 20 out of 80 people, significant pain. 20 out of 80 people, 0.25. Control event rate is 80 out of 100 people, 0.8. Therefore, the relative risk is just divided ER by CER. It's 0.315, and placebo is the control group, if you remember. And absolute risk reduction is control event rate CER minus experimental event rate. So in this case, it's just subtract this from this, and the relative risk reduction is calculated by dividing the absolute risk reduction by the control event rate. So using the above data, relative risk reduction equals to CER minus ER divided by CER. So all these formulas you have to remember. Relative risk is ER by CER. Okay, and then absolute risk reduction is this minus this, and relative risk reduction is this minus this divided by this. Something to remember because it keeps on coming in the exam, and you have to understand the thing as well, like how to calculate each. Like this is very important. The hazard ratio is like relative risk, but it's used when the risk is not constant to time. It's typically used when analyzing survival over time. Okay, next is numbers needed to treat. So the numbers needed to treat is a measure that indicates how many patients would require an intervention to decrease the expected number of negative outcomes by 1, or increase the number of benefits by 1. It's rounded to the next highest whole number, no fraction. A new drug is trialed for the treatment of fibromyalgia. Drug X is given to 500 people with fibromyalgia, and a placebo is given to 450 people with the same condition. After 5 years, 200 people who received drug X had died compared to 225 who received the placebo. What is the number needed to prevent one death? So in this event, control event rate is 225 who died, and then 450 were the people, and experimental drug 200 out of 500 died, so 0.4. So absolute risk reduction is control minus experimental is 0.1, and numbers needed to treat is 1 divided by, so basically 1 divided by the absolute risk reduction is the number needed to treat, is 10. So another example is a drug S is given to 100 smokers to help them quit, while placebo is given to 150 smokers. 60 smokers on a drug S quit smoking, and 60 smokers on placebo quit. So what is an NNT for drug S to help people quit smoking? So control is like 60 out of 150 on the drug S, so it's control. Sorry, on placebo is the control. So 60 out of 150, 0.4, and 60 out of 100 on the drug S, 0.6. So 0.6 minus 0.4. Experimental minus control is absolute risk benefit difference, 0.2. So NNT is 1 divided by 0.2. So NNT is 1 divided by CER minus ER, or absolute risk reduction. NNT of 5 does not mean if you treat 5 people with drug S, only 1 will benefit. It rather means for every 5 people treated with drug S, 1 additional patient would benefit compared to placebo. So yeah. So 1 additional will be benefited if you treat 5 people with drug S. To put this in context, if you treat 5 people with placebo, 2 of them will benefit. If you treat 5 people with drug S, 3 of them will benefit, 1 additional. This is the concept of NNT. So how much numbers you need to treat, like how much much more benefited. Okay, sensitivity and specificity, another important topic. Imagine a scenario where people are tested for disease, and the test outcome can be positive, sick, or negative, healthy. While the actual health status of the person may be different in that setting. True positive, sick people correctly diagnosed as sick. So that's true positive, they are sick and they're diagnosed as sick. False positive is healthy people wrongly identified as sick. So the test is positive, but it's false. True negative is healthy people are correctly identified as healthy. True negative. And false negative, sick people wrongly identified as healthy. Okay. So sensitivity is the number of true positives and divided by number of true positives plus false negatives. So false negative, where we have to go through it again. So true sensitivity. So number of true positive, as in people who are actually sick, diagnosed as sick, divided by number of true positive plus the number of false negatives. That means sick people wrongly identified as healthy. So true positive and true false negative has one common thing: sick people. So sick people divided by sick people. The sick people divided by total sick people. That's the thing. Sensitivity. Maybe positive or negative. So sensitivity. And a sensitivity of 100% means that the test recognizes all sick people as such. Thus, in a high sensitivity test, a negative result is used to rule out the disease. Okay, sensitivity alone does not tell us how well the test predicts other classes. That is about the negative case in the binary classification. So not really necessary, but just read through it. Sensitivity is not the same as positive predictive value. Ratio of true positive to positive. The calculations. So that's how it is. Now specificity: number of true negatives divided by number of true negatives plus false positives. So true negative people are healthy people, which are healthy, and false positive is like also healthy people wrongly identified as sick. So it's like healthy divided by total healthy. So sick people sensitivity, healthy people specificity. Specificity of 100% means that the test recognizes all healthy people as healthy. There's a positive result in a high specificity test is used to confirm the disease, confirming the disease, and this rule out disease. Maximum is trivially achieved by a test that claims everybody healthy regardless of the true condition. Therefore, specificity alone does not tell us how well the test recognizes positive cases. We also need to know the sensitivity of the test to the class, or equivalently the specificity to the other classes. A test with a high specificity has a low type 1 error rate. Specificity is sometimes confused with the precision of the positive predictive value, both of which refer to the fraction of returned positives that are true positives. The distinction is critical when the classes are different sizes. The test with a very high specificity can have a very low precision if they are far more negative than true positive, vice versa. Increasing the cutoff of a positive test result will decrease the number of false positive and hence specificity. So these are the formulas we need to remember. Sensitivity, it's how many of the sick can the test identify, sick versus sick, and how many healthy can the test identify. Specifically, positive predictive value is all positives, all PPP. How many of the test positive samples are actually sick, and how many of the test negative samples are actually healthy. So all negatives. Likelihood ratio of the positive test value, sensitivity divided by 1 specificity, and likelihood ratio for negative result is 1 sensitivity. Positive and negative predictive values are prevalence dependent. Likelihood ratios are not prevalence dependent. So you have to remember all that. That's evidence-based medicine. Evidence-based medicine types of studies: randomized control trial, Participants randomly allocated to intervention or control group, standard treatment or placebo, practical or ethical problems may limit use. Case control studies, patients with a particular condition are identified and matched with controls. Data is then collected on past exposure to a possible causal agent for the condition. Inexpensive, produce quick results, used for studying rare conditions and prone to confounding. Okay, cross-sectional survey provide a snapshot, sometimes called prevalence studies. Provide weak evidence of cause and effect. And cohort study, two or more are selected according to the exposure to a particular agent. And then and followed up to see how many develop a long-term side effect. It's cohort study, long-term. The usual outcome measure is therapeutic risk. For example, the Framingham Heart Study. So the key points you need to find out and write them down. Long-term study, cohort study. RCT, randomized control trial, that's we studied here, involves the random allocation of different interventions, treatments or conditions to subjects as long as the number of subjects are sufficient. Randomization is an effective method for balancing confounding factors within treatment groups. Randomized treatment prevents systemic difference between treatment groups. Cohort study is done for a group of people who share a common characteristic or experience within a defined period, are born, leave school, lose their job, or exposed to a drug or vaccine. There's a group of people who are born on a day or in a particular period, say 948, from a birth cohort. The comparison group may be by the general population from which the cohort is drawn, or maybe another cohort of persons thought to have had little or no exposure to the substance under investigation, but otherwise similar. Alternatively, subgroups within the cohort may be compared with each other. Similar background as well. Case control is a type of epidemiological study design. Case control studies are used to identify factors that may contribute to a medical condition by comparing subjects who have that condition with patients who do not have the condition. Case control. Have the condition, do not have the condition. Cross-sectional studies form a class of research methods that involve observation of some subset of population of items all at the same time, in which groups can be compared at different ages. With respect to independent variables as IQ and memory, the fundamental difference between the cross-sectional and longitudinal studies is that the cross-sectional study takes place at a single point in time, and that a longitudinal study involves a series of measurements taken over a period of time. Both are a type of observational study. So cross-sectional studies are used in most branches of science, in the social science and other fields as well. It takes a slice of the target group and bases its overall finding on the views or behavior. So level and grade of evidence and recommendation. So level 1A, meta-analysis of RCT. 1B, at least one RCT. 2A, at least one well-designed control trial which is not randomized. 2B is at least one well-designed experimental trial. 3 is case, correlation and comparative studies. And 4 is panel of experts. And ABC is basically at least one randomized control trial. B is non-randomized control trial, panel of experts. Okay, new drug studies. So when a new drug is launched, there are a number of options available in terms of study design. One option is a placebo controlled trial. While this may provide robust evidence, it may be considered unethical if established treatments are available and it does not provide a comparison with standard treatment. So if a drug is therefore to be compared with an existing treatment, a statistician will need to decide whether the trial is intended to show superiority, equivalence, or non-inferiority. So superiority will be, while this may be seen the natural aim of a trial, one problem is the large sample size needed to show a significant benefit over existing treatment. So this is the problem, you need a large sample size. Equivalence margin is defined on a specified outcome. If the confidence interval of the difference between the two drugs lies within the equivalence margin, then the drugs may be assumed to have a similar effect. Non-inferiority, like equivalence trial, but only the lower confidence interval needs to lie within the equivalence margin. A small sample size are needed for these trials. Once a drug has been shown to be non-inferior, large studies may be performed to show superiority. So this is the one. You should remember that the drug companies may not necessarily want to show superiority over existing product. If it can be demonstrated that their product is equivalent or non-inferior, they may compete in price or convenience. Intention to treat analysis is a method of analysis for RCT in which all patients randomly assigned to one of the treatments are analyzed together, regardless of whether or not they completed or received that treatment. Intention to treat analysis is done to avoid the effects of crossover and dropout, which may affect the randomization of the treatment groups. Okay. Phases of drug approval. First drug trial in humans. Phase 1. First drug trial in human. Check safety in healthy volunteers in a small group of people of 20 to 80 people to ensure its safety. Determine a safe dosage range and identify side effects. So phase 1 is first drug trial in human. Check safety in healthy volunteers in a small group of people. Small group will be there. To evaluate its safety, determine a safe dosage range and identify safe side effects. Establishing the testing protocol, a drug is given to a larger group of population to see its safety, efficacy, and dose ranging into the target population. Final testing, drug is given to a large group of people, 1,000 to 3,000, to confirm its effectiveness, monitor side effects compared to commonly used treatments, and secure license in target population. So just remember the numbers. Phase 4. Okay, so of course some things need to remember, like phase 1, like safe dose, determine a safe dosage range, safety, side effects, and safety, efficacy, dose ranging in target population. Effectiveness, monitor side effect compared to commonly used treatments, compare. Okay, and post approval studies, post marketing studies usually need additional information with the drug's risk, benefit, and optimal use. So some specific words need to remember: target population, dose ranging, safe dose range, phase 1, and side effect, monitor as well. This one also identify side effect, monitor side effect compared to commonly used treatments, and this one is post marketing studies, risk benefit ratio. Funnel plot is primarily used to demonstrate the existence of publication bias in meta-analysis. Funnel plots are usually drawn with treatment effects on the horizontal axis and study size on the vertical axis. Study size, treatment. A symmetrical inverted funnel shape indicates the publication bias is unlikely. Conversely, an asymmetrical funnel indicates a relationship between the treatment effect and study size. This indicates either publication bias or systemic difference between small and large studies. So symmetrical like this, no bias. Funnel plot showing the publication bias. Each dot represents a study measuring of the effect of certain drug. Y-axis represents study precision, and x-axis difference shows study result. Okay, general medicine, miscellaneous plus oncology.